Hey you guys, welcome to part 2, Notes of Meiosis. So in the last lecture we talked about why meiosis was important because it causes variation. And we talked about why variation was important in a population. And so in this part of the notes we're going to talk about the process of meiosis. So what actually happens to create gametes. And so here, let's do a little review of some vocab. Let me get a draw tool, and here. So what we're doing today is we're talking about how gametes are produced. And remember that gametes are sperm and eggs. So your whole body is produced of body cells or somatic cells or diploid cells. So remember that we are today producing haploid cells. I'm going to put an H over here to represent haploid cells. And the haploid cells are sperms and eggs. And these are different from our body cells because they have half the amount of DNA, or half the number of chromosomes. So I like to remember H for haploid and H for half. And so our sperm and eggs have 23 chromosomes. So 23 and 23. So we go through a process that takes a regular body cell that has 46 chromosomes, and we cut the amount of DNA in half. And so why do we do that? Well, so that when a sperm fertilizes an egg and creates a zygote, well then it's going to have 46 chromosomes. And those 46 chromosomes make it a diploid cell. So at that point, when we have a fertilized egg, here's our zygote, our fertilized egg. At this point, this cell is going to start going through mitosis and dividing and becoming a person. So the point of meiosis is that it's going to have, H-A-L-V-E, the number of chromosomes. It also allows for variation. So it's very important. Let's go to so germ cells are the diploid cells, and they're going to become a sperm or an egg. And so in females, this is meiosis occurs when a girl is still in utero, so before she's even born. Most of the meiosis happens to her before she's even born. So she has ovaries before she's born, and inside of those ovaries are germ cells that are going to become eggs, and those become eggs before she's even born. So most of a female's meiosis occurs before she's even born. Males, however, are going to perform meiosis throughout their lives after puberty, so anytime they need to produce sperm. And so here, interphase starts with a cell, a normal body cell that is in the ovaries of a female or in the testes of a guy, and it's going to start off as a normal body cell or diploid cell that has 46 chromosomes. So let's put a D over here. And so in the process of meiosis, we're going to start with one diploid cell, but end up with 4 haploid cells. And so a germ cell is something that is a cell that's found in the ovaries or in the testes that is eventually, it is a diploid cell that is going to go through meiosis and become either sperm or eggs. So let's look at this process. We're going to go through prophase, metaphase, anaphase, telophase, and then we're going to go through it again. We're going to go through prophase 2, metaphase 2, anaphase 2, telophase 2, and then cytokinesis. So you already know what happens during mitosis, and these processes are very similar. So we're not going to talk about all the things that you already know. Like, for example, you already know that in prophase, the nuclear membrane or the nucleus is breaking down. And you also know that the DNA, let me draw some DNA here, a little from mom and a little from dad, you know that it's starting to coil up, and you know that pretty soon your chromosomes are going to move towards the center of the cell. So you know that that's true. And because you took notes on variation before, you know that this is where crossing over occurs. And if you remember what crossing over is, remember that our chromosomes have homologous pairs. And so your first chromosome from your mom lines up with your first chromosome from your dad. And then during crossing over, what happens is the chromosomes, the homologous chromosomes, line up so tightly around each other that they actually swap DNA. And so here, we're going to just draw that out a little bit. And this is where that's going to happen. So crossing over occurs during prophase 1. So this is in the nucleus, and crossing over is starting to happen. When we get to metaphase 1, what's different about metaphase 1 in meiosis as compared to metaphase in metaphase 1 in meiosis as compared to metaphase in mitosis is that you know that during mitosis the chromosomes line up. You have 46 chromosomes that line up all down one straight row like that. But in meiosis and metaphase 1, our chromosomes are going to line up just like this. They're going to line up with their homologous pair. So you were going to have, instead of 46 chromosomes down, we're going to go 23 chromosomes down, like this. And so we have our homologous pairs. Here's our first chromosome, our second chromosomes, our third chromosomes, all the way down to 23. And what I haven't drawn in here is that these chromosomes are all crossed over. And I'm not going to draw this every time because it's kind of a pain. So I want you to remember that from here on out, every time we draw a chromosome, that it is crossed over. And so this is not just a chromosome from mom, but it's a chromosome half from mom and half from dad. And during metaphase 1, what happens is that the homologous chromosomes assort independently. And this should sound familiar from our last notes, which means that some of the chromosomes from mom are on this side and some are on this side. So it is irrelevant what this pair of chromosomes are doing to these chromosomes. So these chromosomes are going to line up however they want. They're going to be independent of what these guys are doing. So some of mom's are going to be on this side and some of dad's are going to be on this side as well. And that is what's going to happen during metaphase 1, is that the homologous chromosomes line up along the axis, and when they line up, they assort independently of one another. During anaphase, let me move this X so it's not crushing my head. There we go. So during anaphase 1, we know that anaphase apart. We know this from mitosis, anaphase apart. So let's draw our spindle fibers that were growing in our last metaphase, in our last stage. And so here we have our chromosomes are going to start migrating to the sides. Actually, let me erase this because we're going to draw them going this direction. 1, 2, 3, 1, 2, 3. And so in anaphase and mitosis, we had just little halves of our chromosomes migrating to the sides. But here we have entire chromosomes migrating to each side. So here, and remember that they're crossed over. I'm not going to cross them over because it's too tedious and I don't want this to be 30 minutes long. So here we have our chromosomes, entire chromosomes migrate to opposite sides. And here, I'll just cross this one right here. Boom. And this one's got some blue on it as well. So they're all crossed over. So when this cell splits, it's going to have some of mom and some of dad in it. It's going to be something entirely brand new. It's never been created before. So in telophase, we know that during telophase that the nuclear envelope is starting to reform, and we know that the chromosomes are relaxing into chromatin. So we're not going to write any of that down. because we know that from mitosis. So let's just know that this is what's going to happen during telophase. The same thing as mitosis. The only difference is that instead of half chromosomes, we have entire chromosomes. Now, we have two cells. We started with one cell that had 46 chromosomes, and now we have two cells that have 23 chromosomes. So let's put 23 in here. But we said that we're going to end up with 4 cells that have 23 chromosomes. So let's talk about doing this again. We're going to go through meiosis 2. So meiosis 2, remember that usually at the end of mitosis that our one cell that divided into 2 cells would usually be going on into interphase and go through the cell cycle. But this does not happen. No interphase, no DNA replication. That doesn't happen. We go straight from telophase straight into prophase 2. So we know that during prophase that our chromosomes are coiling up and that our nuclear membrane is starting to break down. We know this. So we're not going to write all of this down because you already know this. Something to note that during prophase 2, what happens is that no crossing over happens during prophase 2. That only happens in prophase 1. And that's because homologous chromosomes are lining up next to each other in prophase 1. But that's not happening in prophase 2. Let's look at what the chromosomes are doing as we move towards metaphase 2. So metaphase 2, our chromosomes, 23 chromosomes in each cell, but we're going to just make this 3 chromosomes in each just to make it easier. So we have 3. They're going to line up along the axis. So here, and you can see that each of these cells are going to be different because the chromosomes are sorted independently. And we know that metaphase, the spindle fibers begin to form. Here we go. Our spindle fibers are forming. Here we go. And here, here, and here, and then here, here, and here. So this is what's happening during metaphase 2. Our chromosomes are lining up, and now they're going to be just one chromosome. There's no more homologous chromosomes. Our homologous chromosomes split into 2 different cells. So in this cell, we have one first chromosome, one second chromosome, one third chromosome. And then in this cell, we have one first chromosome, one second chromosome, one third chromosome. We know anaphase apart, anaphase apart. This is when our chromosomes are going to start splitting up. So let me make some really short little spindle fibers. Boom, boom, boom. And boom, boom, boom. And here, here, and here. And now our chromosomes are going to, just like they did in mitosis, now this is going to look a little bit similar. They're going to split in half. The sister chromatids are going to split in half. And they're going to start migrating to the sides. And let me get the blue here and here, and we're almost done. And you can see how the cell right here is also starting to split into 2. Anaphase of 2, our chromosomes are starting to migrate to the poles. And telophase 2, we're almost at the end. You can see now that we have 4 different nuclei, and they all have we know that the nuclear membrane is reforming and the chromosomes are relaxing into chromatin, and we now have 4 cells that each have 23 chromosomes. We now have, we have one cell that have 4 nuclei that have 23 chromosomes. So 23 here, 23 here, 23, here, and 23 here. 23 chromosomes in each. So this is one cell that's going to split into 4 haploid cells. Here we are on our last slide. So during cytokinesis, our cells split. And for females, 4 eggs are formed right here. 1, 2, 3, 4. And this actually has a pretty cool name. It goes through a process called oogenesis, where 4 eggs are formed. And actually what happens is one egg gets most of the cytoplasm. It's going to be bigger and it's going to be stronger. And the other 3 are called polar bodies, and they die off. But they all have 23 chromosomes. So we have 4 haploid cells. I'm going to put 4 H cells, 4 haploid cells. And for males, 4 sperm are produced. So 23 chromosomes here, 23 here, 23 here, 23 here. So 4 haploid cells are formed. And this is important that they have half the amount of DNA as a regular body cell, so that when a sperm fertilizes an egg, it is going to be able to have the appropriate 46 chromosomes. And each of these cells are completely different than from who they're coming from. It is made from who they're coming from, but it is all of the DNA is recombined. And so whoever whatever their offspring is, we know that their offspring is going to be different from them. And that allows for variation. Okay, that is that. I hope you guys have a great day, and let me know if you have any questions.